in this episode of the Critical Oxygen Podcast.
Heat-shock proteins are like housekeeping proteins that we have in our cells, and they're in charge to pretty much to like support or help out with the folding of newly synthesized protein. That's pretty much kind of be like the major role that they do inside of the cell, right? Everything from proteins in our hair, in our muscle cells, in our skin cells, in our red blood cells, the hemoglobin of our red blood cells, our white blood cells, everything is made up of proteins. So it's very important that proteins maintain a specific shape and form in order to function effectively. If for some reasons proteins unfold or lose that shape, they're also going to lose their function. So they're not going to be working effectively, right?
Welcome to the Critical Oxygen Podcast, where we help you optimize your physiology and maximize your endurance potential. I'm your host, Phil Batterson. I have a PhD in molecular exercise physiology, and I'm the founder of Critical Oxygen, where my mission is to build better endurance athletes through remote education and in-person physiological testing. And today I'm joined by Dr. Maria Fuentes, where we're going to talk about heat shock proteins and their role in exercise and disease. Dr. Fuentes is a U.S. ECFMG Certified Medical Doctor and graduated with a Master in Public Health from the University of Texas at El Paso in 2018. She was born and raised in El Paso, Texas, and Maria has focused her research studies and gained experience on different areas of medicine and public health that affect the health of the community of both El Paso and Juarez, Mexico, including infectious diseases and environmental health. Maria is now pursuing her PhD in Interdisciplinary Health Sciences at UTEP, studying the effects of climate change, such as heat waves, on the expression of heat shock proteins, like HSP27 and 70, and that's good. We're going to focus a lot on these today, and their associations with kidney injuries. She is currently a full-time faculty member at the Department of Biology at El Paso Community College, teaching biology, anatomy, and physiology courses, as well as a guest lecturer at the Department of Public Health at UTEP, teaching courses in statistics and public health evaluation. Whew. That is a long resume. That's going to be hard for a lot of the other guests coming on the show to live up to, Dr. Fuentes. So thank you so much for coming on the show.
Well, thank you so much, Phil, for having me. I have to say I'm a big fan of your show. And like I was telling you right now, I always learn so much from your podcast and each one of the guests that you have. So it's a pleasure for me to be with you today.
Yeah, I appreciate it. If I remember correctly, I think at some point I had talked about, or mentioned, heat shock proteins or some level of cellular or molecular regulation, on the podcast or, in an Instagram post. And you reached out to me and you're like, hey, that's actually what I'm studying. And I was like, great, let's get you on the podcast like right now. And you're like, no. I'm kind of busy, doing this thing called, like getting ready. What were you were proposing, right? Your PhD?
I just presented, defended my proposal like just 2 weeks ago. So I'm just fresh out of the oven.
Yeah. Well, I am hoping to take full advantage of all that preparation that you did, for your proposal and everything like that, because that is a massive step in the PhD journey. So congratulations on that.
Thank you so much.
And I imagine it was a little stressful because. Most of the time,
definitely it was. I just wanted it to be over. I'm like, that's it, please. I don't want any more, but we're done.
Yeah, but I mean, being a medical doctor and other things like that, I'm sure you've done a lot of studying in terms of boards and becoming a certified medical doctor. So, it's one of those things where in the moment it's really tough, but I'm sure you had the tools to get through it.
Yeah, of course. I remember you were talking about heat and the impact. I think you had a guest and you guys were talking about the effects on heat, on exercise, and you brought something up about heat shock proteins. I'm like, hey, I'm so glad that somebody's bringing up that topic. Yeah. So because it's kind of like a topic that is kind of like beginning to like you hear about it on research and all of these important aspects. So I was glad to hear about it.
Yeah.
That you bring it up.
Yeah, and the interesting thing, so this is a caveat for all the listeners out there, is that our conversation is going to be more of an exercise in biomolecular physiology. It's going to be talking about how heat shock proteins kind of interact with the body and the signaling mechanisms that are going on. So be prepared for some pretty deep physiology, which is exactly what I love to talk about. But from and hopefully we'll be able to take a step back and learn, from an applied perspective. But really, this is a knowledge journey for all of us here. So I just want to give that caveat. But before we actually jump into heat shock proteins and their signaling mechanisms and those sort of things, Dr. Fuentes, if you wouldn't mind just giving us a little bit of background, how did you go from, like medical doctor, master's in public health, and now a PhD? Like what were the things that aligned to bring you on that journey?
I think when I came here to the US, I was studying the boards. I was doing a lot of practice, mostly the pediatric field. And I didn't spend too much time, like honestly, on clinical practice, maybe 4 or 5 years. And I was trying to get ready to get into the residency. But to be honest, I don't know if this sounds a little bit gonna be sounding a little bit bad, but I noticed that in clinical practice, it was going over the same things over and over and over. So I was like going all through medical school to be there looking at the same repetitive cases from 8 to 5. It was kind of like a big important like moment for me because I didn't enjoy so much clinical practice. Don't get me wrong, I loved studying, going to medical school, and I guess that's where we get like the nerdy side of it, right? Like understanding how everything works, trying to get an answer for everything of physiology, diseases, everything. I wanted to understand everything in medicine. So I love that part, like the studying part. But clinical, I found it a little bit challenging and not so much enjoyable for me. So, but throughout the practice, I did notice patterns like in childhood obesity, metabolic syndrome, a lot of in children already, like kids going into elementary, middle school, already showing signs of metabolic syndrome, a lot of behavioral disorders, so many things. And also, for example, including the aspect of antibiotic resistance, infections. So for me, it was kind of like patterns of diseases, of things that I was looking at in medical practice. So I got interested in, can I do something more about it? So that's how pretty much I got into population, I guess, medicine, which it will be public health. And I got into research I went back to school and got my master's in public health and actually focused on infectious diseases and antibiotic resistance. And trying to understand how different environmental factors play a big role and everything, like connecting that we are developing all this big problem with antibiotic resistance as well. So from there, I guess I just decided to continue. I love research. I love, I guess, this is what it gets exciting about research, right? There's always like something new that you do not know, that there's a big challenge that you want to figure out that maybe you can do in research, you can study it. So I just decided to continue and go into academia. So I just love being part of academia and finding all these new things that I just do not know and do not have an answer. So that's like the big challenge for me. So I just love studying and getting into these issues, public health issues, I guess.
That's awesome. We definitely need that your type of thinking, especially when it comes to medical practices and stuff. Like you were noticing all of these things that we're doing the same treatment over and over and over again, and perhaps there's a better way to potentially go about that. So
I felt like I was going one patient at a time, and I was just not getting anywhere. So I guess more research is needed, especially here on the border. We require like a lot of input and research, especially on the Hispanic population. So that's where I moved.
Awesome. Well, that sounds like that's a very awesome career path for you. And who knows, going forward, it's like I'm the same type of way where, I get passionate about, a certain area of things. And then, then my focus changes. And, like everyone always asks, oh, in 5 years, what do you think? Because I have no idea.
Exactly.
Like something else is going to come along that's going to be super interesting to me. So it'll be fun I'll definitely be following your work, going forward to see how things go.
Thank you so much.
Yeah. So I would be remiss to say that this is an exercise physiology, bent podcast. So, do you have a background in exercise science, sport, or anything like that? And do you use like the knowledge from your physiology to maybe shape your research questions or vice versa?
I guess in my profession, my background, like educational, I do not have like any expertise on exercise physiology. Of course, I just have the knowledge, my medical expertise, and what I know from my PhD, but it's not my area of focus. I guess I'm just simply a runner. I love running, and I try to apply like all the knowledge and understand absolutely everything, right? If I have to do zone two, why do I need to do zone two exactly? Yeah. I have to have like a purpose and an understanding for it to like really like understand about it. Sometimes I go and talk to other like running groups and I guess share a little bit of my knowledge and a little bit of the little that I know, of course, and I just go ahead and share that information with them, so we can understand, become like better um, performers, better at the sport that we love, and be able to do things with a purpose. So,
yeah, I'm very similar in the way that whenever I'm doing something, whether it's from the academia side of things, from a business side of things, from a training side of things, I really like to understand the why behind it. It's like, why are we doing zone two? Why is it beneficial for us? Why is high intensity interval training beneficial for us? So it's interesting to see that those interests and the things you're trying to explain, go from like the medical profession all the way trickle down to you said I'm just a runner. It's like, well, it seems like you're thinking about this stuff on a deeper level than a lot of people, normally do.
Yeah. Well, of course, like I was saying, I like to understand everything, how the muscle cells, they're working. Why do I need to slow down? Why need to I bring this? Why the feeling is important at different levels, right?
Mm-hmm.
So, I guess I just have to understand that in order to make sure that I apply it correctly. So,
yeah, and one of the things that you said to me is like, well, like I was asking Maria offline, oh, so like, let's talk about, like heat shock proteins and their relationship to muscle physiology. Do you have some reviews to send me? She's like, I'm more of a kidney, physiologist, but you sent me a really good review article, which I'll put in the description down below for those of you who want to read it. It's all about heat shock proteins and their relationship with muscle and exercise, and not necessarily just muscle, but related to exercise. So, if you wouldn't mind, let's start jumping into this. Let's go ahead
and jump and talk about it. Let's go into it.
So first and foremost, can you kind of give us like a definition of what heat shock proteins are? When I hear heat shock proteins, the first thing I think about is, oh, these things are related to high temperatures, like heat shock, right? What we're going to find out, I think, without spoilers, is that it's actually just a stress response protein. So let's hear from like from the expert's mouth, what are heat shock proteins and kind of what are, well, and then we'll go from there. So what are they? And then we'll talk about the function.
Okay, so I'm gonna go talk a little bit like the ABCs of biology. I'm gonna go like and try to go like the most simple way that I can, right? So pretty much heat shock proteins are like housekeeping proteins that we have in our cells, and they're in charge to pretty much to like support or help out with the folding of newly synthesized protein. That's pretty much kind of be like the major role that they do inside of the cell. Now, in order to understand this, we have to go back, right, and do, like I was saying, the ABCs of proteins. So let's go ahead and take a step back and talk about the cell. We have the nucleus, and in the nucleus, we're going to go ahead and bring out the central dogma of biology, right? And here we have to talk that in our nucleus, we have all these DNA and genetic information, and all DNA, you have all of these genes, right? Like thousands of genes that we have, and they're pretty much the purpose of it is kind of like a big recipe of life to synthesize and to make new proteins, right? Because everything, like I tell my students, right? Everything from proteins in our hair, in our muscle cells, in our skin cells, in our red blood cells, the hemoglobin of our red blood cells, our white blood cells, everything is made up of proteins. So it's very important that proteins maintain a specific shape and form in order to function effectively. If for some reasons proteins unfold or lose that shape, they're also going to lose their function. So they're not going to be working effectively, right? So again, going back, right, we have the DNA that making to the messenger RNA. It's going to exit the nucleus, and then finally it's going to go into the ribosomes, right, where we're going to have the primary sequence of proteins, which is going to be made only of amino acid sequences, right? So all of these primary sequence of proteins is going to have to go into endoplasmic reticulum, and right there into the lumen of this endoplasmic reticulum, we have a lot of these molecular chaperones, which are the heat chain proteins. Now, these guys are going to be in charge. They're going to just pretty much like supervising the work. There are other proteins. They're being folded like properly into secondary structures and finally get that final 3D structure and become like functional and have that proper function, right? And after that, well, they're going to go into the Golgi apparatus where they're probably going to get the signaling and their shipping address, right? Are they going to stay inside the cell? Are they going to become part of the plasma membrane? Or are they going to exit the cell and become like somewhere pretty much like somewhere else? I always tell my students, you know, like when you order something from Amazon, right? They don't send you the product like that, right? They have to pretty much maybe put it in a nice bubble wrap, like, you know, put it in a box and then go ahead and ship it to your address. So it's going to be the same thing of the protein. They have to be folded properly and go through that process. And all of this is going to be the assistance of heat chain proteins. So it's very important to understand that the heat chain proteins, they're going to be working inside of the cell, making sure that not only the new synthesized proteins maintain their shape, but however, when the cell is exposed to any type of cellular stress, right? And this can be heat, it can be a state of hypoxemia, ischemia, it can be metabolic stress, right? For example, like in running, like metabolic demands of intense aerobic exercise. It can be also like, for example, exposure to toxins, medications. So anything that can actually trigger some type of stress to the cell is going to start activating these heat chain proteins. Now, why? Proteins, if we'll go back again, right, to ABCs of biology, they're very susceptible to two, primary responses in the environment, right? One is going to be temperature and the other one is going to be pH. And any changes of temperature, pretty much proteins have to be ideally to maintain that structure between like optimum levels of 37 degrees Celsius, pretty much between 35 and 40. So proteins are going to be working effectively, hence our normal temperature, right? To work effectively within those that specific range. Now, when there are like going to be any type of specific stresses, in this case heat, right, it's going to start like activating heat chain proteins, because proteins, they can start losing that shape, become denatured, they become like loose. A lot of these hydrogen bonds and other type of bonds that pretty much provide like the shape of the protein, they start to break under these heat insults or stresses, and they break apart, so they lose the shape. So heat chain proteins are going to be patrolling, you know, like within the cell and making sure that all of the proteins, not just the newly synthesized ones, but the ones that are already like there within the cell, maintain that cellular integrity. Now, when there's a lot also like too much damage within the cell, heat chain proteins are actually in charge to prevent cells going to apoptosis and going to cellular death, right? So they play a big role actually in maintaining that normal homeostasis and the normal, function of the cell. So they have like a very protective mechanism within the cell, and they're like overprotective. They're making sure that the integrity of the cell, it's there, and everybody's in their shape, in proper shape, so they can function effectively. However, I guess they can be like very susceptible, I guess, to trigger that response. For example, in exercise, I just noticed, right, that even I guess we're gonna have to talk a little bit later on about thermoregulation to talk about heat stress and all of that. But as short as we start producing heat, And even as 38.5 can actually trigger the heat shock response, like through exercise, right? Because as we start kind of like producing heat when we're working out, they're actually like working to make sure that to maintain that normal homeostasis within the cell, within the skeletal muscle cell as well. But in cases, I guess, of heat stress, like if you're working out outside and it's very hot and all of that, I guess a response, like a normal triggering response, it will be within 38, 40 that is already activating this heat shock protein mechanism. And you can stop me and like anytime I can just keep going.
You're putting on a clinic in terms of explaining all of this stuff really amazingly. Something that I did want to just summarize in general is that it's not only heat that is activating heat shock proteins. It's really any sort of cellular stress. So just to summarize that, it's these heat shock proteins are reacting to cellular stresses, and like you said, some of the main ones are heat and pH changes. We know in the muscle can get really, really warm when we start to exercise. And the problem with that is essentially you're cooking yourself from the inside out. So when we talk about denaturing proteins, it's essentially like you put an egg in a frying pan and, the egg white starts to firm up. That's almost what we're doing, right? And the problem is that in the body or in our body, when things start to get warm, we're actually affecting the function of things like mitochondrial electron transfer proteins, or the ability to maintain sodium-potassium gradients, or ATP, or calcium, or all of these different things. And if we don't have highly sensitive regulatory mechanisms to stress, bad things are going to happen when we start to exercise, right?
Exactly.
So the heat shock proteins, like you said, are kind of like the you said like kind of the manager of the construction company. They're making sure that like the people underneath them don't screw it up, and then on top of it, they're also managing like the other building projects that are going on over here, and they're saying, okay, well, we got to make sure that the building is actually staying intact, because if this building starts to fall apart,
Exactly.
That's bad. So they're the foreman of the construction contractors within the cell, and they really play a role in proteostasis, which means that your protein levels, or your proteins, are staying in some level of steady state or homeostasis. They're being built correctly, they're being maintained correctly. And if they aren't maintained correctly, then heat shock proteins can, for kind of a crude way of describing it, they can cause degradation. So if they see in the building, oh, that door is rotting out or that door is broken, they can say, hey, we need people, we need our contractors to go fix that door. So it's absolutely fascinating, and it seems like there's actually a lot more roles than just simply being like a signaling mechanism back and forth. But that is the major role, is proteostasis, and kind of being an intermediary in terms of communication of the stress that's going on.
Yeah, I think they're the ones that are gonna try to maintain as much as possible to prevent your cell into apoptosis and try to patch things up the best possible way and protect your cells the most that they can. Now heat shock proteins, they can be classified into molecular sizes. We have the small ones, the 27, for example. Then we have the 40s, the 60s, the 70s, 90s, and we can go to the large ones up to 110. Usually the one that has been the more spent time I guess doing research on is going to be the 70. I think heat shock proteins, the first time they started to arise in research was around the 90s, but I think it's been really kind of like the last 10 years where they started to picking up to really understand like their functions and their important role not only thermoregulation, but actually the protective role in maintaining this cell integrity and homeostasis as well. Now, I guess about the 70 is going to be the constitutive form. That's going to be the normal one. So the 70 is always going to be forming like a complex with a heat shock protein transcription factor, which is the HSF1, and they're going to be always like bond together. Now, when they're like exposed to any type of stress that we already mentioned, they're going to kind of like release from each other so they can start going again into the nucleus and start like upregulating genes. In this case, it's going to be the 72 is going to be the one that is the isoform that is upregulated from the 70.
And this would be HSF would be released from that kind of complex. Exactly. It would go into the nucleus, bind to the promoter regions of the different DNA or the heat shock protein 70 DNA, and probably some other ones have more mRNA of heat shock protein 72, right?
Yes, correct.
Because there's 27 and 72, and I'm like, do I have dyslexia or something like that? But no, they're it's the size. So it's 27 kilodaltons and 72 kilodaltons. And so it's a little confusing when you read it.
And it's funny that you mentioned that, because in my research proposal, I'm using both 27 and 72. Yeah. And somebody tried kind of like bring up like the error. I'm like, I think you put 27 instead of 72. I'm like, no, I'm using also 27, as a control heat shock proteins to notice like the difference in expression in this case. But yeah.
Yeah. It's always so interesting in like how things get named. Well, we won't even go down that rabbit hole because some of the stuff just doesn't make any sense. But
in this case, it's at molecular weight. So
yes,
it's gonna be
so the 27 is the molecular rate. The heat shock portion of it was probably some they noticed that due to increases in heat, these proteins were released. And I think back a little while, like a few years ago, people started talking about these, and they were like, oh, you have to go into the sauna to get your heat shock proteins up, and then these heat shock proteins are released into circulation. And people were making conclusions, I think, based off of data that wasn't there yet. Because we still don't really know, and we'll talk about this in just a little bit, but we still don't really know why heat shock proteins make it into the blood and what their function is then, other than probably being it's probably like saying, hey, there's a fire, and then signaling to the other areas of the body. But we still don't know how then do those heat shock proteins go to the brain? Do they go to the kidney? Do they have different binding domains on different cells in order to upregulate things like different cellular processes? But most likely they're probably being released in order to be able to help maintain some level of steady state or homeostasis, which I always talk to people about. Is that's what your body is trying to do in response to exercise. Is we are trying to do everything we possibly can to reestablish some level of steady state or homeostasis during exercise. So most likely heat shock proteins are doing that.
Yeah, and right now that you mention it, it's very important, I guess, to make that big difference. When heat shock proteins are inside of the cell, they have this protective role within the cell. Now, they are anti-inflammatory, they do their own work, but when these heat shock proteins actually start leaking out into the circulation and you find them in plasma, right? Actually, they're already sending a danger signal, like, say, something is going on here, and they actually become like starting activating the immune system. So they have like an opposite effect, and they can activate both. They can activate the innate immune system through like toll-like receptors and start like activating that immune signaling cascade, but they can actually go the adaptive, which is the most common one, the adaptive immune response, start activating through your major histocompatibility complex 1 and start activating your CD8, your T cells, and start like activating this whole inflammatory cascade of everything. The only issue that I see and that I have in research, right, is that they're not like very specific to what organ, like you find them in plasma, like you cannot really tell like where they're leaking out, like from what type of tissue, which is kind of like that issue, like to pretty much to be able to detect. Now there's like a lot of research showing histopathological changes when take biopsies and they see the expression of these HITCH proteins, like to even like changes within even 20-30 minutes of stresses like occurring within the cell, right? But when they start leaking out, I think that's going to be the problem, because they start activating all this whole immune reaction and cascade of inflammation. So
yeah, the contractor goes from being like, okay, we have everything under control, to kind of pulling all the fire alarms, right? You know, saying
exactly.
everybody something bad's happening. And I guess it kind of makes sense if you know, they trigger a little bit or an immune response, they may be reacting to fever, right? Heat shock
Exactly. Yeah.
you know, like while I kind of gave it a hard time saying it shouldn't just be heat shock proteins, but there is probably some response to high core body temperature that results in heat shock proteins being released that can then be stimulatory of the immune system and then cause more of that systemic response.
Yeah, exactly correct. So, um, I guess in this case, for example, the kidneys. I have to talk about the kidneys
Of course.
because, of course, kidneys are my areas, right? So we have the nephrons, right? The nephrons is gonna be the primary cell of the kidney, right? And you're gonna be able to find different type of HITCH proteins against along the different parts of the nephrons. But the one that you tend to find throughout the nephron, the different segments of the nephron, is going to be the HSP70. Now, in this case of the kidney, for example, they're not so susceptible like to like changes in heat, but the reaction that actually is going to trigger is going to be changes in renal osmolality. In these cases, for example, the changes that you have like in sodium that is found in plasma. So these changes actually in osmolality, like the sodium concentration, is the one that is going to start activating. And you can actually, well, there's studies that you can actually start like finding these urinary HITCH proteins after this type of insult. So that's why I wanted to actually focus my research in runners, because, you know, we're always running out there in the sun, changes, and we're not doing this hydration properly, right? There are changes in the cardiovascular, in heart rate variability, and then changes in the sodium concentration. And of course, I think they're perfect to study them. But right now, it's not my area of research. But I think they're like the perfect participants to study this expression.
Yeah, I was just, as you started to talk about that, I was thinking about how could something like dehydration
Exactly.
start to trigger, right, heat shock protein response within the kidneys. And you know, especially exercise in the heat, right? Your core body temperature is going to increase, but one of the ramifications is that you sweat more, so you lose plasma volume, which can change osmolality of you know, the blood. What about this is just kind of an aside, but what about things like type 2 diabetes? Did those change osmolality of the blood, and then can those activate heat shock proteins as well?
Yeah, I was reading some studies on diabetics that they have like an impact on this renal osmolality, and they have been able to find these HITCH proteins in patients like with diabetes, or um, because I mean a lot of these patients with diabetes tend to develop these chronic um, disorders, affectations in the kidney. So there's like an affectation, and yes, there's like an expression of this in these tissues.
Yeah, and I always think about stress from a time-based perspective. When you exercise, I'm using stress kind of loosely here because stress is really hard to actually define. But when you exercise, you have, you know, an increase in energetic demand, a change in redox balance, like a change in calcium kinetics and homeostasis and other things like that. Temperature goes up. You might have, you know, some level of uh, decreased oxygen availability, and all of these, you know, they act, and it's really it can be a really massive stress on the body. But then if you are in a healthy state, it goes back to baseline or steady state over the course of the next few hours as you recover and other things like that. But in terms of pathophysiologies like type 2 diabetes, which can be factors from, like having too much adipose tissue on your body and stuff, you actually wind up creating a stress on the body that is lower in absolute magnitude, but it's chronic over time. So you start to extend this quote unquote stress for days to weeks to months to years. And then that's where systems start to fall apart, right? If you're always activating, say, heat shock proteins, for example, in response to, high blood glucose, eventually, well, potentially, that response could be blunted or start to go away or start to not be as potent of a de-stressor as it once was. And I think you said earlier that heat shock proteins are at least their functionality or their ability to maintain proteostasis actually decreases with pathophysiology. So I'm wondering if there's some level of regulatory mechanisms that are going on within those pathophysiologies that are actually inhibiting the ability of heat shock proteins to depress the stress. So maybe you have, constitutively active, low level inflammation because heat shock proteins are activating your inflammatory response via, the immune cells in the immune system. And that just doesn't ever go away, right?
Yeah, exactly. And because what is not really known, because even though they trigger like this inflammatory response, like some heat shock proteins do release some cytokines that are not, they're protective. Actually, they decrease the immune response. So it's not very specific to which heat shock proteins elevate, like, which create like this immune reaction, and which kind of like go and try to like slow down, like, like slow down the fire. So I think in that sense, there's still like a lot of research that needs to be like to really done. However, I think there's a lot of going on in to see that to be able to use them as potential biomarkers for diagnostic purposes and as well as for treatment purposes. I think they're trying to understand like this microenvironment, for example, that occurs like in cancer cells to actually to develop these, I guess, targeted therapeutic approaches using heat shock proteins. But I think in that sense, I think we're getting started, but there's like a big gap of research that we can do right there, and big opportunities that we can do over there, like in that area.
Yeah, it's in the research world, when there are huge gaps in the literature in terms of pathophysiologies, generally, that's where money can be found and research can be done.
Yeah, exactly.
The tough thing is that like people who are passionate about exercise and all of that, it's to some extent it's kind of hard to monetize exercise and get people to exercise. It's a lot easier to monetize a pill, by doing something like, giving people heat shock protein therapy or inducing heat shock proteins or, something along those lines. Right. So, it's just the way it is in terms of research, but it sounds like, there's a lot of questions that still need to be answered here, which, for a budding researcher in this field is, pretty promising, right?
I find it exciting. I love the topic. I think it's like so fascinating. It's I don't know, you can see like climate effect, like climate science. You can study like the medical aspect of it, therapeutic approaches, physiology, just the molecular like understanding of them. I think it's a beautiful topic. But for example, if we talk about exercise, right? Since like all of your audience mostly exercise, and like I think we brought it up like in the beginning, right? I was just reading the expression of these heat shock proteins, for example, 72, and the important role that they have in the tapering process. So after you go all of this intensive training, right, that we have to taper, and we have to understand why tapering like so important, right? Well, heat shock proteins are like kind of like reconstruct, like doing this healing process in your skeletal muscle fibers, right? And it seems like a lot of these performance in work they're doing, mostly I think on type 1 fibers, they're pretty much making sure that you're ready for for
Mm-hmm.
that race. So I was surprised to see that they're beginning to understand the role even on this type of exercise, right?
Yeah. Let's jump into that. We've kind of talked all the way around. But in terms of exercise and stuff, there I think we'll approach it from first an acute perspective, and then we can kind of jump into like what happens after training, and then the get to the tapering portion as well. But how how does the expression of heat shock proteins change in response to an acute bout of exercise?
I was reading about it. Like I'm saying, it's not really my area in
Mm-hmm.
exercise, but actually, even like 1 hour of intense aerobic exercise, because and also including that metabolic demands that is like you're putting the cell with the needs of glucose and fluid filling and all of this, because we're talking about anything can be a stress to the cell that is requiring some type of adaptations within the cell is going to trigger the response of a heat shock protein
Mm-hmm.
within the cell. So you can start like noticing the elevation or the expression of these proteins in as short as 1 hour
Yeah.
of exercise, intense
Yeah,
exercise training.
generally what we see when we're looking at expression of either proteins or mRNAs that are related to proteins is that a single bout of exercise, um, it's pretty time course dependent, but a single bout of exercise will induce increases to mRNAs of these proteins, and heat shock proteins are no different. They're maybe even reacting even more quickly than a lot of other proteins. So you have a spike in mRNA, and then not all of that mRNA gets translated into proteins. So eventually, over time, you start to actually build up. I think you start to build up protein amounts of different populations of HSP70 and potentially other ones, but really the 70 starts to accumulate, and it seems like that it's related to our ability to fend off future stresses. So if we have more of those heat shock protein chaperones around, we have more monitors of, like our construction site. And if our construction site can be more well monitored, then we can signal and we deviate a little bit less from homeostasis each time we exercise. So it's really interesting that in the skeletal muscle, you see this increase in mRNA immediately following exercise, and then you see a gradual build of these heat shock proteins over time in response to your exercise training. And then another interesting thing is that you get a release of these heat shock proteins into your blood as well. And we've talked about this too, but it seems like not only like that's when like those heat or the heat shock proteins change from the chaperone to the signaling molecule, right? They change into a little bit of a cytokine. I want to call it a myokine just because it's like, oh well, it could be expressed from the muscle, but it doesn't seem like that's the case. It seems like it's more kidneys, heart, potentially brain, that are expressing heat shock proteins into the blood. And again, I think that's a little bit more of like could be potentially the systemic stress response, which is then trying to signal to other tissues in the body, hey, we need to be more robust next time we do this. So, let's put some into the blood, and then it'll eventually go away. But it's that acute upregulation and then building over time, that could be helping to make us more stress resistant. Like, I
Yes,
think that's
exactly.
ultimately, yeah.
With it, like helping with that process of adaptation. And I was reading that their expression tends to be like higher in the type 1 fibers.
Hmm.
So I guess that's why like they're like so important in endurance training.
Right. That and from a muscle physiology perspective, I think that makes sense, right? Because type 1 fibers are generally more fatigue resistant. And if we have more protectors of stress, then we have a greater tolerance for stress within that cell. Obviously, depends probably on the exercise intensity and other things like that. But it seems like, you know, to build on top of things, these heat shock proteins are also, muscle fiber specific, And one thing that was really interesting to me is that when they overexpressed, I think it was HSP70 in skeletal muscle of mice, they showed a more mitochondrial heavy phenotype. And, if you ask anybody, hey, what's the master regulator of mitochondria? They would say PGC1 alpha. But it seems like heat shock proteins also play a role in this. And I don't know if it's a direct interaction with, say, PGC1 alpha, which is a transcription factor for regulating mitochondrial biogenesis or mitochondrial, just form and function in general, or if it's a divergent pathway that's also important for it. So someone would have to knock out PGC1, and then they would have to upregulate HSP70 and see, do you still get that, same response? And another thing that they said is that when you downregulate heat shock protein 70, you get these like weirdly shaped dysfunctional mitochondria. And I was telling you offline, I applied for a grant during my PhD that wanted to look at the unfolded protein response. So when your proteins get damaged, they become unfolded, like exactly like what you're talking about. They get denatured. And if you don't have mechanisms to trigger degradation, then you don't get rid of the bad proteins. So you get this increase in proteins that aren't functioning correctly, and then ultimately that results in poor functioning of whatever organelle those proteins are in. So it's just, that was a lot.
And this is exactly the role of the heat shock protein. Like they don't only help with this the new proteins, they also like assist with preventing these unwanted protein aggregation. Exactly what you're talking about, right? They have like this big role like in that sense. But I was also surprised like to see their big contribution to mitochondrial biogenesis, and I was like, whoa, so they also help out with that. So I'm telling you, it's amazing, and it makes sense. I mean, with when we're talking about this the type of metabolic demands that we put the skeletal muscle in this type of exercise, right? So like as short as 38.5 can activate heat shock proteins, and of course, like we're producing a lot of heat when we're working out. Also, like the needs, the fueling need, like glycolytic needs, the oxygen needs, right? So it's interesting to see like all the role that they play in all this activity.
And we're not, we're saying like we're all of these crazy roles, and the crazy thing is we still don't know exactly what they're doing. Like the molecular pathways have not been fully mapped out for how these heat shock proteins actually are say contributing to mitochondrial biogenesis, or contributing to mitophagy, because that's the sequestering and breakdown of mitochondrial membranes that aren't functioning correctly anymore. But I will say this: if heat shock proteins are activated through proton or increases in acidity, and if you have mitochondrial membranes that aren't working correctly anymore, they're most likely leaking out or exposing more protons to the cellular environment. So if these heat shock proteins are able to chaperone autophagy markers or other things like that to that area, which I think they did say like BIP, which is, I'm pretty sure, related to this is like 2 years into my PhD, and it's been a long time since then. But there's some chaperoning of like those mitophagy markers, or I'll say protein degradation markers, because they might not be specifically to mitophagy, but just autophagy in general, or cell cleanup. So it seems like they're helping on both sides. And if you think about what we need to do in response to exercise, is we need to manage and make new proteins, and we need to manage and get rid of old proteins. So if heat shock proteins are kind of in the middle, that fulcrum of protein synthesis and protein degradation, then it makes sense that they're going to be involved in all of these different things. And like you said, there's the constitutively available HSP70, but then there's ones all the way from 27 kilodaltons to 130 kilodaltons, and we still haven't mapped out everything that's going on with all of them.
And I think that the reason that they study like the 70, 72 so much is because like the 72 is a one that is highly inducible to, especially to heat.
Mm-hmm.
So that's the one that is like really rapid, shows like a response and being like upregulated. So I think that's why like they tend to focus a lot of the research on the 70. So I think it's easier for research purposes to track and to like mark the expression levels, like
Mm-hmm.
in that sense.
Yeah, and that's generally how it starts, right? And I think too with all the new omics approaches, proteomics, transcriptomics, all of that sort of stuff, we're going to start to be able to identify more novel proteins that then what we do in research with like omics techniques is we cast this really wide net and we say, okay, what groups of proteins, what types of proteins are changed based on a certain intervention. And then you can pick groups, right? The heat shock proteins probably being a really good group for this. And you can say, okay, well, I know that within this group of, heat shock proteins, there's 20 different proteins, something like that. This is all hypothetical stuff. But then you can say, okay, I want to focus on this one, heat shock protein 70. And that's probably like, to some extent, that's somebody identified it. They said, okay, this seems like a really good marker for all these different things. Let's see what happens when we put it into mice and upregulate it, when we put it into mice and downregulate it, when we expose mice to heat, when we expose mice to cold, when we expose mice to metabolic stresses, all of this sort of stuff. And it's something that's amazing about like basic science. And I say it's called basic science, but it's not basic. It's actually really, really complicated stuff to do. And it's really awesome because what you can do is you can systematically go through and say, is this protein necessary for this function? And there's a lot of amazing people who have done, like careers worth of work on, just specific proteins or protein complexes. There's this one guy actually at this mitochondrial conference who has systematically gone through and knocked out every single protein of the mitochondrial complex 1. And I can't remember exactly how many proteins are in mitochondrial complex 1, but it's like, 20 to 40. So he has systematically gone through and knocked out and seen what has happened to complex 1 and its respiration in response to it. And I'm just like, oh my gosh.
Wow, like now that's a dedication to really answer that question, right?
Yeah. But it's just absolutely fascinating to me. And it seems like we're just at the tip of the iceberg with our understanding of heat shock proteins. Cause even that review you sent that I'll post in the description below, there's some really good foundational studies that are out there, but there's not very many.
I think so far, like we know the expression, it's there with the histopathological levels, but still there's more to explore to understand, for example, what happens when they start leaking out of the cell, like to understand this like immunoregulation, this immune response, and also how can we are able to identify them if we can use them as biomarkers.
I wanted to ask you about that, the biomarker idea. So so obviously you must have some idea of what we think they could be doing as biomarkers, but what can you share with us about that?
For example, right now, talking a little bit about kidneys, right? For example, in case of heat stroke, right? I think patients who undergo or like develop heat stroke, about 40% of patients develop acute kidney injuries. And from those, up to 80% have like a fatality, end up like with dying, right, of a heat stroke. So, but the kidney is one of the primary organs that tends to be damaged when you have like that heat, when you're exposed to heat, in the case of a heat stroke. Now, the issue with identifying, like in clinical like diagnosis with acute kidney injuries is we currently still use the serum creatinine, which is like the most common biomarker that we use in clinical practice to develop to pretty much to see how your kidneys are functioning.
Mm-hmm.
The only problem with the serum creatinine and the bone is that pretty much we don't know if the damage that you have in the kidney it's acute or it's like a chronic effect. Because, for example, let's just say that you had a chronic disorder, right? Or like diabetes, and you already had some damage of your kidneys.
Mm-hmm.
So it will not be able to let us know if this renal damage was because of recent exposure to heat, right?
Mm-hmm.
So, and also, for example, when you have like this heat stroke, the kidneys start producing serum creatinine up to maybe 48 hours later, so we can pretty much start treating your kidneys accordingly, like in that case. So we're losing 2 days of pressure time, like to be able to determine if your kidneys were damaged because heat-related illnesses. Now, with the heat shock proteins, for example, in case we know they're expressed, right? Like a lot of research is supporting that information. But however, we know that they're expressed within even 24 hours, way before the serum creatinine. So they seem to be like a favorite candidate among like all the researchers that we can have like this potential use of using heat shock proteins as biomarkers of acute kidney injuries in cases of heat stroke. Right now, we're having like an epidemic of acute kidney injuries in those that are working outside and they're like chronically exposed to heat. For example, construction workers, those in agriculture, they're constantly like exposed to high level temperatures, right? And all of this population, they're having like a big impact, and we have an epidemic of chronic kidney disorders because of this heat exposure. That's why right now this population we're trying to focus on trying to identify specific biomarkers that if a person comes to emergency department, we're able to pretty much detect an acute biomarker so we can treat your kidneys accordingly. The good thing about acute detecting an acute disorder is that it's reversible. Pretty much, you treat it and it's a reversible process. But most of these, when they become like unattended, they develop into chronic kidney disorders, and most of these patients tend to have dialysis. Now, those patients that have like acute kidney injuries or anything, right? When they start also releasing this serum creatinine, which is the current biomarker, usually when you start having symptoms of kidney diseases, it's already like the damage of your nephrons. It's too late. Sometimes you can have like up to 50% of nephrons damage like in your kidneys to barely start showing symptoms. So it's already too late to start treating them accordingly. That's why there's like an urgent need to start like finding possible biomarkers that can give us like a quick interpretation of an acute damage, and we we can rescue and treat it accordingly. So right now these heat shock proteins are kind of like the most favorite candidate among all of those that are doing research on kidneys and these heat shock proteins as biomarkers.
So in terms of damaging the kidneys based on heat stroke and other things like that, is heat exposure but not getting to the level of heat stroke, is that an effective way of inducing a stress response in your kidneys that make them more tolerant to heat? Or is it just kind of one of those things where your kidneys are generally pretty fine up into a certain temperature, but once you start to get into that more like heat stroke sort of thing, too much dehydration, like those sort of things, then it's kind of like a oh boy, this isn't good for our kidneys? Because I imagine a lot of people listening to this are like, oh my gosh, I'm being exposed to the heat every single day by doing, a lot of exercise in the heat and stuff like that. Are they causing potentially long-term damage to their kidneys because of the heat exposure that they're experiencing?
Oh, that's interesting question, but that's kind of like a long, like a long response on a long topic, right? I guess we have to go back to the basic mechanisms, right? But no, in that case, we have the process of thermal regulation and thermal, a process of adaptation, right?
Okay.
We adapt little by little to this heat exposure and exercise. That again, the heat shock proteins help us through all these adaptation processes to maintain that integrity, just in a nutshell, right? To maintain that integrity with this exposure. I guess when we're talking about heat, right, we have to understand that when we talk about heat, we don't have that blunt, just 37, boom, that's our main temperature and that's it. We're like constantly fluctuating. And this is where we have like this amazing, highly evolved thermoregulation response, right? We have, I guess we have to talk about, we have two different types of temperatures, right? We have the shell one that is going to be kind of like our skin, right?
Mm-hmm.
Everything, all the receptors that we have like in our skin. Then we have the core temperature. And those are going to be always like fluctuating between, I guess, 36.5 and the 30, like before the 37 something, right? We're like we're constantly fluctuating within that temperature, right? So and I guess with that adaptation process, these heat shock proteins help us as we're constantly exposed and we're running as well and being constantly exposed to heat.
Mm-hmm. Yeah, it's always just, there's always a fine line between, like exercising and accumulating enough stress and then having it go overboard and being like, well, that was too much stress and now I'm injured or now I'm sick or now I've, induced damage that, may not be the most appropriate, for exercise adaptations and other things like that. But I'm
and
glad.
I guess I'm so sorry. And I guess that's why it's like so important to like when we're like, especially like here in El Paso, right? It's so important to adapt your paces and your running to according to heat and stay, please, for the love of God, I always say, right? Stay hydrated properly with the proper hydration and sodium and all of that so you can function, effectively. The good thing is that, for example, the heart rate variability is like such a good indicator when we're going through this process of adaptation, right? When we're running like in the heat, right? And I guess I'm going to throw the question back to you, right? So if you're, let's just say that you're running in the heat, you have like the perfect control, your heart rate is perfectly, and you start like having this heart rate variability at the end, right? Because we're not like keeping up with the constant fluid intake and the sodium and electrolytes by the end of a run, especially if it's hot. Now, I guess my question back to you will be, do I need to slow down, like to be able to bring down and keep down my that heart rate where it was like at the beginning, or is it okay like to like to have that change kind of like at the end of that workout?
That's a great question. I wouldn't say I don't use heart rate variability per se, but I do use heart rate, right, to train, especially like in the heat. And what I will say to people is that, if you're doing, say, like your zone 2 workouts, right, it should be fairly maintainable for extended periods of time. You go out and, you got 2 hours or something like that of zone 2 training. It's a little warm, right? It's super hot well, I say super hot. It's nowhere near how warm it is in El Paso, I imagine. But I have an idea of in a controlled environment, like in the room that I have my treadmill and my bike, typically I can maintain, say, a heart rate of 120 to 130 for really, really long times. I've been doing more heat training lately where I just say, okay, I know the power output that would elicit 130 beats per minute. So I'm going to just kind of focus on that power output, and my heart rate over the course of an hour, hour and a half, maybe even 2 hours, will rise from 130 to like 150 or 155. That's an indication to me that my body is having a challenge getting enough blood to my muscles and getting enough blood to the skin to cool. That's still well within my general, like maximal heart rate's like 180 for me. So I would say if you're trying to get heat adaptations, obviously, like consult your doctor, consult a physician before going out and exercising in the heat. But generally, if you're maintaining a controlled intensity, it's okay to have your heart rate rise. Just know that following that, you're probably going to be more dehydrated than you normally are. So drink more fluids. Make sure you are refueling properly, because when you're exercising in a higher stress environment, you have a tendency to be more reliant on things like carbohydrates. So you're going to want to refuel with that. And from a hydration perspective, it's still tough to say whether you should be intaking like a lot of electrolytes or a moderate amount of electrolytes or other things like that. If you eat a diet that has a lot of added salt and sodium, then you probably don't need to supplement with it. But if you're making a lot of your food and you're not adding salt to it, then it won't hurt. Again, consult your physician before doing any of this. It won't hurt to supplement with some level of, sodium and other electrolytes. And if you start feeling like it's always better to be conservative in terms of like the type of exercise you're doing in the heat. So if it starts to feel hot, it's always better to back off than to keep pushing. To keep pushing, you're running the risk of developing heat stroke, and then you're going to be a candidate for Dr. Fuentes' clinical trials. But we don't want you to be doing that to yourself. So be conservative with your exercise, but it is okay that your heart rate actually comes up. That was a long-winded answer to basically say that.
But I think, I think it's also important to say that actually anyone can be exposed to heat stroke, because I think even those that are like we're adopted, it's just like heat stroke. I think we gotta be able to make a difference, right, between heat exhaustion and heat stroke, because in heat stroke is just there's just like a loss of control of your thermoregulation process, like, and your body's just gonna completely go start like producing a temperature over 40, and pretty much you're gonna start like maybe developing like all of your organs can actually be affected in the case of heat stroke. But I actually think that like anybody can be exposed to heat stroke. In that case, maybe heat exhaustion, it will be more like you will develop like more adaptive mechanisms to prevent heat exhaustion. But heat stroke, it will be just anyone can actually, your body just loses that complete control of being to maintain that thermoregulatory process.
Okay, so your definition of like heat exhaustion is, because your physiology has to work harder to maintain blood flow to the skin, blood flow to the muscle. Heat exhaustion is more of like the fatigue you feel from being in the heat and other things like that. Whereas heat stroke is actually like your body no longer has the ability to regulate temperature effectively. So
exactly.
we almost like almost anybody who's exposing themselves to heat is doing some level of heat exhaustion in a sense, but we never want it to get to the point of heat stroke. And I also remember too reading something where individuals who experience heat stroke once are more susceptible to heat stroke following that. Is there any validity to that?
I cannot really back that information up.
Okay.
Like I haven't I cannot maybe don't have information to back that up. But I guess when we're talking about heat related illnesses, there's just like many of them, and it can go from something as simple as a heat cramp, for example, and those are like very common that you see a runner pretty much running in the heat and they start experiencing some type of heat cramps. So that's like one that's very common, right? There's also like a heat rash, and that's very, for example, common in babies, because their sebaceous glands, they kind of like become clumped, and you kind of like see these miliaria, right? This kind of like rash that babies tend to develop. Then we have, for example, like heat exhaustion. Like in this case, of course, you start producing a lot, your temperature starts kind of like raising up. You become a little bit confused, disoriented, very dehydrated. Like you kind of like have like those symptoms that you're not feeling okay. You're dehydrated. Maybe you're sweating a lot. You're sweating. You're actually producing sweat. In the process, for example, of heat stroke, you're not producing, that mechanism is not being effective. So your body actually becomes like warm over 40 and you're not producing sweat.
Yeah.
So that mechanism also becomes disruptive. So that's like very completely different, because in the other one you can start developing seizure, develop coma, and even a lot of people kind of like, we have like a high mortality of heat stroke. So those are like very completely different mechanisms of heat related illnesses.
Yeah. It's heat's a scary thing. It's, I always tell people, like, I personally prefer cold because I can put more clothes on, but as you get hotter and hotter and hotter, there's only so much you can do to cool yourself. So be smart about it, everybody. If you have any questions, you can of course ask me, and I'll point you in the right direction to good resources in order to kind of avoid that sort of stuff. So we've kind of gone over on time and everything, Dr. Fuentes. I've really been enjoying this conversation. So with a couple minutes left, what, from an application point of view, if there are any, what do you want, people to walk away from this conversation with, whether it's heat shock proteins, just heat mitigation, other things like that? What's the big takeaway here?
I guess it will be hard to focus on heat shock proteins, right? But I guess for us, it will be to understand that there's a lot of physiological adaptations happening in our body, and maybe that's why in terms of exercise, that's why so important to understand like the different phases that we have in training, right? And understand that a lot, maybe we don't see big changes and big effects that you're having during training, right? But we have to be able to know that at molecular level, there's a lot of things happening, even when you're taking a rest day, when you're taking a recovery run, when you're tapering, right? There's a lot of things that is happening at molecular level, like in ourselves, to help out, to have a great performance and a great outcome at the end. So I guess from an exercise perspective is, for me, will always be respect the purpose of each workout, right? Because at molecular level, something is helping you out, something is happening. I guess from a heat point stand of view, it will be, be respectful to your environment. You adapt your paces to the heat, and of course, stay hydrated. Please, protect yourself. And if you have any questions, go ahead and visit your physician about any of these maybe imbalances, to make sure that you're taking care of yourself and training properly.
Yeah. I love what you said there. And I think, just to bring it back to heat shock proteins, they're really quite interesting in terms of the very large potential roles that they have on the adaptive process within the body, as well as stress mitigation. So this is absolutely fascinating. I'm hoping to get more people who, are very, specific in their types of research and stuff like that. So if anybody out there who's listening to this is either an expert in a certain type of field, it doesn't have to be, perfectly exercise phys related. We'll still have an awesome conversation about things. Let me know. And I would love to get you on the show. That's kind of how Dr. Fuentes and I, started out and everything like that. So this is a really awesome conversation. I really appreciate you coming on. Where can people find you? Is there like an Instagram or anything like that? ResearchGate, like those sort of things?
I do have ResearchGate. My Instagram, honestly, is just I'm just having fun with my running journey.
Okay.
It's my running progress and me having fun. You can follow me at Dr. Fuentes_runs, but it's just me having fun. I don't have anything educational or anything formal, in that sense. And you can find me, I guess, on ResearchGate as well as Maria Fuentes.
Yeah. If you are interested in Dr. Fuentes' research and those sort of things, I highly recommend signing up for ResearchGate. It's a way, if you haven't heard of it, it's a way of potentially interacting with researchers, getting papers from those researchers, being able to read peer-reviewed literature in a way that isn't just, relying on Google Scholar to figure out, what you're supposed to be reading. So
and the
head
cool
over there.
thing about ResearchGate, I'm so sorry, is that you can read the papers even before they're being published.
Yes.
So I think that's a kind of like the big point of ResearchGate.
And sometimes you can, if you don't have access to a journal that would require you to pay for a paper, you can get it directly from the author, and you're not breaking any rules doing that. So, head over there if you're interested in learning more about heat check proteins and what she's doing. As always, you guys can find me on Instagram at Critical02. Let me know, like if you know people who are, kind of into the molecular side of things, I'd love to get them on. I love talking about this sort of stuff. If you have any questions, comments, you can comment down below. If you're on YouTube, you can comment. If you're on Spotify, if you've been enjoying this podcast, please leave a 5-star review on Spotify. It really helps me out and gets more exposure to, kind of the mission of helping people understand the physiology so they can, either maximize endurance potential or maximize health and wellness. So I hope you guys enjoyed this conversation with Dr. Maria Fuentes all about heat check proteins, and we'll catch you guys in the next one.